And this is, the title of the presentation is Annotation and Comparative Analysis of Insulin-like Peptides and Their Interacting Genes Across Drosophila? Drosophila species. Okay, that's good. And I'm not even going to pretend to say anything that I know about this. The floor is yours, and we're excited to hear what you have to say.
Does this work?
It's a good question. We spent 15 minutes just getting this to project, so
Okay.
let's see.
All right,
It would
that's
work on
okay.
the computer actually, so if you point there, but I'm not sure.
That's okay.
I can run it for you if you like, if that would be easier.
Oh, no, I got it.
Okay, you got it? Okay.
All right, so hello everyone. My name is Logan Cohen. I'm a biology major, and I'm going to talk about fruit flies. Specifically, the annotation and comparative analysis of insulin-like peptides across and their interaction genes across Drosophila species. So before I get into that, I'd like to give special thanks to my faculty advisor, Dr. Darren Barnard, and I'd also like to give special thanks to the Genomics Education Partnership for providing the database and other tools that were necessary to complete this research. So this is Drosophila in the 7 groups. These are the 7 species that I used in my analysis, and we're going to focus in on 2 of them. But what we're going to go over is first insulin-like peptides and the potential relationship between those proteins to the phenotype or external appearance and chemical nature of the fruit fly. And then we're going to go over an interspecies and intraspecies comparison of ILP receptors. And then we're going to trace these changes in the insulin signaling network all the way through the cell using the growth modulation pathway. So what is an ILP? When we think about insulin, we think about diabetes, which is the dysfunction of sugar metabolism in the human body. Drosophila have 8 different ILPs that we know of in the insulin signaling network. And all of them are triggered at different life phases with different environmental triggers. ILPs control things like sleep-wake cycle, adiposity, gastrointestinal function, reproductive function, and even the transition from one life stage into the next, because Drosophila have several of them. So, you spot the difference between these two? You can shout it out. Size. Yep. And
Yeah.
I wonder if it's a different color. You're right.
Yeah, because one is a bumblebee and one is a slide.
Yeah, they look very different. They're completely
Yep.
different species. So these are the 2 species that I'm going to be focusing in on, which is Drosophila hydae, which is the larger, darker fruit fly, and Drosophila melanogaster, which is the smaller and more yellow one. And I chose these species specifically because they represented 2 different subgenuses in the Drosophila genus. So this is a multiple sequence alignment of ILP6. So what on earth is this? This is an alignment that shows with ILP6, which is the ILP that allows for growth under starvation conditions in development. The only areas between these species that are conserved are the domains and some areas that are immediately on the periphery of the domain. So domains are the regions of a protein that protein needs in order to fulfill its role. Outside of these domains, there's a little bit of flexibility for evolutionary change, but all of the domains here were conserved. But despite that, there was a lot of difference in that periphery outside of the domains. So when I saw that, my first thought was, well, that's not supposed to happen, because we expect the insulin signaling network to be very well conserved. This is a network that we see across the animal kingdom, including humans, and also I'm looking at the same genus. So why, even though I'm looking at different species, is there such a wide variation in this peptide? So I started with tracing it into the cell to see how far these changes would go. There's 2 receptors for insulin-like peptides. The first is INR, that is the active receptor. What happens when an ILP attaches onto the receptor? It sets up Chico, which is a signal protein that sets off a cascade of signaling throughout the cell. SDR is a decoy, it is a down regulator of this system. So SDR binds to ILP and no signal is sent to the cell. So I was expecting to see very minimal difference between the 2, maybe just enough to explain how these ILPs are able to interact with these very different signaling networks. So I went and I looked and I saw, okay, how similar are these within the species? And what I found was very consistent. When we look at these two these two receptors in Drosophila melanogaster, we see 18.2% similarity. When we look at it with Drosophila hydae, we see 18.4. That is very, very close, kind of impressive for something that's across species. But when I look at them between species, it became a lot more apparent that there was a lot of change happening even on this level in the network. So comparing INR with Drosophila hydae, which is the dark one, and melanogaster, which is the light one, I saw 59.1% peptide similarity between the two. And for SGR, it was a little bit better, we have 73.8%, but that is still a pretty low amount of similarity in peptides for something that should otherwise be very well conserved. So I decided to trace this through 3 different pathways, and the one that I'm going to show you today is the growth modulation pathway. So the way this one works is ILP2, again there's 8 of them, they're very creatively named 1 through 8. Attaches to INR, goes through Chico, there's a few other genes in between there, and then we end up with IMPL2, which produces the peptides that control and modulate growth. So what I saw with this was with ILP2 between Drosophila hydae and melanogaster, and this was very consistent actually across the phylogeny. It was very, very low rate of conservation. There's 56.6% similarity between these two peptides. So going into the network with INR, that's the receptor, 59.1% similarity. This is very consistent. And then Chico also had a 58.1% similarity between these two. What I was expecting was to hit a wall of basically conservation somewhere shortly after Chico. But the reason why the intermediary genes are omitted is because this pattern just went on and on and on and on until that final gene, that IMPL2. IMPL2 had an 83.3% similarity between these two species, which seems pretty impressive and like we're approaching the rate of conservation we expect in this network. Except that the isoform and exon structures in these species were completely different. So in this chart, I'm showing you the 2 species that we looked at up close, Drosophila hydae and melanogaster, but also Drosophila busky. And I included that one because that is in the 3rd subgenus that I was working on in that project, and it's a very good reference. So these fruit flies are colored as they would be in real life and are also to scale. You can see the difference in size between the two, with Drosophila hydae being almost double the size of Drosophila busky. And what we see here in this region, this is what genes look like if you're looking at them in any sort of software model, where we have an intron structure. These are the things that are spliced out before we go to peptide production. And then these exons here, which are the parts of the gene that actually make the protein, which is the whole point of having genes in the first place. So what I saw was that in Drosophila melanogaster, we had 3 different isoforms. Drosophila busky had 1, which was roughly analogous to one of the ones that we find in Drosophila melanogaster. Drosophila hydae had 2, and also had a different exon structure. Again, with all of these, the domain regions were conserved, but that was about it. And I went through and did some confirmations of these annotations, because when I see something like this, my first thought is, oh, that can't be right. But using some peptide analysis, using synteny analysis, and other means of tracking conservation, it turned out that yes, there is this much of a difference in a gene pathway that is so critical to life as growth modulation. So all of this brought me to a question that I didn't have when I started this, which was, is the evolutionary paradigm incomplete? So by this I don't mean a challenge to the gradual and random mutations over time plus selection model. We know that to be true. What I'm wondering is if there's not something more to that, a more concerted and adaptive method of evolution that allows more homogenized changes within these critical networks over time. I looked into it a little bit, and back in the late 90s, this was a pretty popular hypothesis, but at the time, we did not have the technology to do analysis on this level. And over the last few years, there's been a few papers that have come out in other species, especially in insects, that suggest that there's some sort of homogenized and concerted method of evolution happening in addition to the other methods that we know to already be established. And specifically, I'm wondering, in this well-conserved network, the changes seem to be the greatest in insulin-like peptides. So are these insulin-like peptides mediators of evolution or a loci of evolution within that network? So some questions that come to that, because I did not reach the answer I was looking for with this research. I just ended up with more questions. Could we induce ILPs under shock or stress conditions, and will that generate inheritable change in Drosophila species? There was one paper I saw that was very interesting that suggested that Drosophila were responding as a genus quicker to the stresses of climate change than many other species and even many other insects. And that got me wondering if there's other networks, well-conserved ones, that have trigger genes like ILPs that can provide sort of a locus of evolution that allows for rapid or adaptive speciation. And the big question, because we always want to bring this back to, okay, so what's the point? These fruit flies, they're fun, they're cool, I like them, I don't expect anyone else to like them. In what other organisms do we see these mechanics? Do we have networks like this that can allow for speciation in other animals and also in humans? And by studying those, can we find ways to use novel and upcoming therapies in order to treat things like diabetes, which is really, at the end of the day, diabetes is an autoimmune disorder. It's a dysfunction of the immune system that leads to attacking the pancreas, and then you end up with the final outcome of that, which is problems with mediating sugar metabolism in the body. So anyway, ILPs. I just think they're neat. I would like to give special thanks to my son and my husband over here who have listened to me talk incessantly about this for a very long time. And again, I also want to thank my faculty advisor, Dr. Darren Barnard, and also the Genomics Education Partnership for providing materials for this research. So with that, I'm ready for questions. So there's actually some question of whether or not there's also a diabetes type 3. There's a lot that we don't understand yet about diabetes and why it comes up. There's the autoimmune hypothesis, and then there's these two categories that we have of type 1 or type 2. But the more that gets researched, the more we're finding out they're very similar in their final effect, even though their onset is different. So the application for this with diabetes would be, we're looking at these insulin-like peptides that can all bind to the same receptor. And there's a lot of issues with insulin as we have it, where the storage conditions are very rigid. And over time, what happens is the sulfide bonds in these insulin, in the insulin that we're using therapeutically, you end up with what's basically a prion, because these insulins are binding to each other. So there's a lot of long-term effects that we really don't know. Like, so things like cognitive decline and diabetes, we know that at least a part of that is related to, the damage that happens over time with these wildly fluctuating sugar levels. But I also have questions about, is the way that we're treating diabetes and the problem with that medication related to part of that cognitive decline? Are we basically, by extending lifespan and treating the disorder, are we causing prion disease in people who have insulin-dependent diabetes? Because the cognitive outcomes for folks who are not on insulin and are wildly different. So by studying these ILPs, we might be able to come up with a different mode of insulin, or it's like a different variant of insulin that we could use to treat humans that might have a lower risk profile.
Interesting. I learned a lot. Thank you.
Yes?
I'm not a scientist. How did you do this research? Were you just like combing databases?
Oh, I love this question.
Yeah.
So, my first research semester, I was assigned a gene, and it was Chico in Drosophila hydae. And what I found in that gene was that there were two parts of this gene that were completely dissimilar, as in unrecognizable from melanogaster. So I want to take this back to the tree to kind of make a little bit more sense. There we go. So I started all the way up there on the top with Drosophila hydae. Drosophila melanogaster is in another region. So we expect to see a change in these genes, but not that much. So what I did is I did a process of annotation, which is when you locate where that gene is within the genome of that species. And then once I was able to locate and identify the parameters of where is this gene and what different versions of this gene exist in any given species. I went through and I started comparing where in the network are these things working, where are these genes interacting with each other. And it's kind of like a puzzle where you have these pieces and you're trying to figure out how do they fit together and what does it mean. So did that answer your question at all? Because I can,
No.
it didn't answer your question?
Yeah.
Wait, did or didn't?
It did not. Like, were you, like, how did you do this? Were you like in a lab, like a test tube? Were you like,
So
like,
this
were
is,
you just like combing databases to like look at models, the thing?
Right.
Like what, how did that?
okay, so, yeah, so this is, there's wet benches and then there's bioinformatics. So bioinformatics is how we take all of this data that's accumulated at these wet benches where they're actually doing the DNA sequencing and all of that other critical work, and then we put it on computers. And what that allows us to do is do things like, let's say, Drosophila hydae. Someone on a wet bench, they sequenced that, and then that was taken and put into a database. What you can do once you have that is you can do different types of searches using a tool called BLAST. And BLAST is like the Google of genomics. So one of the first steps of annotation is I say, okay, we know all sorts of stuff about melanogaster, because 110 years ago, someone decided to start studying it, and we have studied it ever since then. So we know a lot. And by pulling the peptide sequence from melanogaster, I can then take that peptide sequence, toss it into BLAST and go, okay, where is this peptide, if it exists at all, in this other species? And then from there, it's a process of looking at some data about where in the genome is their expression, looking for consistencies, like we want the intron and exon structure, or at least like you can think of it like the framework of this particular gene. Do we see consistency there? And then after that, there's other checks that you can do. So synteny is a way of explaining where is this gene in the genome. So we expect when we see a gene that its neighbors are going to be the same from species to species. And that's one of the ways that you can sort of check your work and say, okay, there's a lot of change, but are we even looking in the right place? And if you are, and you have that like genomic neighborhood, and then you also have other markers of similarity, then you can say, yep, this is my gene. And then you get to the bigger picture of, okay, what is it?
Great, that did answer my question.
Okay,
Thank you.
awesome. Yes.
Is there any evidence for alternative splicing with IMPL2?
Yes.
And did you take that into account in your alignments where you would, if you have alternative splicing, you would end up with different isoforms of proteins?
Yes. So what I found was, and I can go back to that graphic. So with isoforms, that's just another phrase for alternative splicing. So with all of these, the domain structures were conserved in all of these. But the difference was for some of these, so this is where the domains lived in melanogaster. In this exon, that's where the domains lived in Drosophila busby. Drosophila hydae was really interesting because instead of seeing the conservation of that one exon structure for these domain regions, it was actually split into two separate exons there. And that brought up questions of, okay, am I seeing a gap there, seeing an intron where there isn't one? But all of the evidence with the expression data, the length of the intron, suggested that, yep, this is another form of alternative splicing.
I think.
Oh,
Last-
sorry,
yeah,
last question, it'll be
yeah.
real quick. Have you considered using artificial intelligence such as classification or association to maybe see if there's certain, I guess, attributes or things about the species that cause the insulin production?
Like that that are really, so,
Yeah. What I'm trying to say is I noticed on your evolutionary paradigm that you have species that branch off. My project is on artificial intelligence, and so you can cluster these species together if they're similar. Do you think that if you can find similar traits that cause the insulin production, that maybe you can predict new species that haven't been discovered?
that could be possible. The part that wouldn't be compatible with AI is that when these adaptations happen, they happen in response to something in the environment.
Okay.
So we could make some predictions about how we suppose a Drosophila would react to, let's say, extreme heat shock or starvation conditions. But until we have the full picture, we can't really know what that response is going to look like genomically. What we can do with AI though is we can, once we start seeing patterns, we can write up algorithms that can look at an entire genome and predict with some level of certainty, there's already some algorithms, some are better than others, what is going on in this particular section of the genome, and those can be used as supporting evidence. So we're not at the point with AI where we can have computers do this annotation for us, but we definitely already have AI support.
Thank you so much. Thanks to each of our presenters.